everyone, I am Odelia. I am honored to present a bit of my research here. So the sense of smell is very important in our daily life. Just think when you smell a perfume or when you get aware of a fire smell. Some people are losing their ability to identify odors. Did anyone here lose this sense of smell during COVID-19? Yeah, I did too. So we will all agree that it is very annoying, and usually it comes back and everything's all right. But unfortunately, it's also one of the first signs of many brain diseases like Parkinson and Alzheimer's. That's why researchers would like to understand much more about this sense. But its headquarters is a tiny part of the brain called the olfactory bulb, which is very hard to visualize with all the imaging methods. Actually, with an MRI scanner, it's possible to get the anatomical image of the olfactory bulb, like you see here in the picture. But what about the activity of the olfactory bulb to be able to, like, how can we get it? So in general, like, that's general concept. So how can we get activity in the brain? So we usually use functional MRI. Functional MRI, so the principle is like we take one picture, one anatomical image. It can take a very long time without limitation, except time is money. And then we have to take a bunch of rapid imaging when you are doing an action. For example, when you tap your hand, you stop, tap your hand, you stop, watching at something, or anything similar. And then we do superposition of all these images, and we can get a contrast that highlights the activity in your brain. So that's general, but for the specific olfactory bulb, it is another story. Why? Because in the first end, the olfactory bulb is a very, very tiny part of the brain and need a high resolution even to be able to see it with an MRI. And the other thing is, like, it is located close to the eyes and the air cavity here, and it causes image distortions. What's image distortions? It's like a stick into a cup of water which looks broken when you take a picture. In MRI, this is these distortions here. You see here? So this will be in this area. So it is a challenging project. So I have good news and bad news. The good news is, like, in the Weizmann Institute, we are lucky to have very high resolution scanners, a 3 Tesla and a 7 Tesla. Just for your information, in clinics and hospital, you just have usually you have a 0.3 Tesla or a 1.5 Tesla. Or, you know, in very advanced clinics, you have 3 Tesla, but it's hard to find. So we can get the good anatomical images. But the bad news is, like, with the current available method in the scanner, we cannot extract the activity information. So here comes my research. So in our lab, we have developed for 10 years a new imaging method called SPEN here. And then as common in research, we must develop protocols and methods in preclinical research here in mice before testing on humans. So we use I developed experimental and processing on getting the activity of the sense of smell in mice, and using a 15.2 Tesla scanner available also in the Weizmann, at the Weizmann. So how does it work? So we take a mouse. This is the olfactory bulb of the mouse here. We put a mouse into the scanner here. We put the mouse. And we also control and monitor the temperature, respiration, and many other parameters. And then we get the anatomy of the olfactory bulb. Here, we can clearly distinguish the brain of the mouse, here and here and here, the eyes, the air cavity, and the olfactory bulb, which is here, and circled in white. So, but how does it work in processing, in real lab life? So what we do, we send air without odor to the mouse. Then we take one picture of the olfactory bulb with our method rapid imaging. We select the olfactory bulb, and then we plot the intensity with, showed with a blue arrow. And then 2 seconds after, we do it again with a pink, showed with a pink arrow. And then we send air with odor through a channel to the mouse. We do the same, and then we plot an arrow. And then this is the raw data, actually, this is the real data. So you can appreciate that the signal increased drastically here. So we have signal during the odor simulation. And then we repeat the process, and repeat the process, and alternatively sending air, odor, and then we get the signal. At the end, we take all these images, and we process them, and we can generate a statistical map. So we did this experiment multiple times with different odors, with fox odor, banana odor, and flower odor, causes a reaction in mice of negative, neutral, and positive feelings in mice. And then we tried also different sex. We tried so many different experiments. The paradigm, what you see, air, odor, air with different times, and so on. And then we discovered this. This study has been published in a report, a scientific report in Nature. So we discovered that different odor stimulation show different patterns into the olfactory bulb. And then, what does it mean? It means that it's also different type of cells into the olfactory bulb. And that's very important to understand and to see it. So now, to the question, is it possible to capture the sense of smell in mice? So the answer is, like, we succeeded with mice, and thanks to its success with our method, we have started clinical research on a 7 Tesla MRI scanner with the same methods on humans. And this helps a better understanding of the sense of smell and might lead to new hopes for early detection of many neurodegenerative diseases. Thank you for your attention.